and we'll get started. I think other people will probably join us, but in order to keep us on time, I want to welcome everybody to our fourth Centers in Iron and Heme seminar series. And this morning, we're going to take a moment to hear from Cindy Roy, who is a program director at NIDDK, to tell us about opportunity funding. And we're going to let her talk first, and then we'll come back to me before I introduce our speaker today. Okay, Cindy. Okay. Thank you so much.
Thank you, Diane. Yeah, thanks for the invitation to be here. And I hope that I caught quite a few of you at the East to West Iron Club a couple weeks ago, which was a great meeting. Again, my name is Cindy Roy. I'm program director in hematology at NIDDK, and I co-lead that program with Shilpa Hattangadi, who has just joined us from Yale School of Medicine. Many people are probably familiar with her work. And we work closely with colleagues like Padma Maruvada, who many of you may know, over in the Division of Digestive Diseases and Nutrition. And so I'm grateful to take just a few minutes here at the beginning to remind you of the NIDDK research mission. As we are an institute that doesn't have blood in our name, I like to remind folks that we are in fact charged by the federal government to support hematologic research. We have just closed out fiscal year 2020, and we have been able to fund about $57 million in hematology research over the last year. And this graphic that you can generate yourself from NIH Reporter shows you that we do have a strong interest in iron metabolism and in heme biosynthesis in our portfolio. Most of that sort of filters into our understanding of erythroid cells, but if your science is leading you in other directions, again, I'd welcome you to reach out to me, and I can share with you some of the areas within NIDDK or the NIH that your work might be better suited. And we do like to highlight some of the great publications that have come out this year. I know that many of you have contributed things that aren't even on this list, but we do take great pride in the work that comes out of the portfolio that is supported by NIDDK. Dr. Griffin Rogers has several core principles that he aims to move forward for the hematology community and for NIDDK at large. And the first of those is to maintain a vigorous investigator-initiated research portfolio. What this means, especially for hematology, is that upwards of 70% of the dollars that go out of our program are for R01s and research programs that you, the investigators, have initiated. We believe that our science is some of the best at the NIH and certainly in the country, because you all are pushing forward the ideas and the new areas that need to be developed within this particular realm. We also want to promote a diverse pool of talented new investigators. And we do that for early career folks, but also for trainees and those of you who are mentoring trainees. NIDDK has one of the broadest programs for training at the NIH in terms of the types of awards that we can offer. And with this program, we can support folks from really their high school years all the way through junior faculty, whether you're an MD, a Ph.D., both, whether your degree is maybe a little bit different than either of those, we can still support your work. One of the ways that we can support high school and undergraduates is through an R25 program and through diversity supplements. So the vast majority of you who hold NIDDK awards that start with an R are probably eligible for a diversity supplement. And then as you move on to doctoral work and postdoctoral work, we would also like to walk alongside of you. And then in the sort of the stages where you're transitioning into faculty and you know, your own independent research program, we have a bonus for folks who are in within 10 years of their residency or their terminal degree. I think for many of us, the need to improve the diversity of our research program is certainly on our minds. And so we have ways that we can do that. I mentioned the supplements to promote diversity. This is an award that is intended for one particular participant, and this allows us to recruit new talent into our research programs across the US. But we also have opportunities in the form of the F31. We have 2 flavors of this award. And so for those of you who might be from underrepresented backgrounds, please feel free to reach out to me and I can provide new and other information for you. But the program announcement is there on the slide for you. And then you all are aware that the Center for Iron and Heme Disorders is one of our NIDDK hematology centers. This is one of our largest programs in hematology to support research nationally. And it provides you all with these great enrichment opportunities to hear from leaders around the country and really around the world. But we also have opportunities to access pilot funds, and this center at Utah is right in the middle of that. And so please continue to look out for opportunities throughout the year to access that program. I know that one of their opportunities has just closed. And then finally, I would just like to remind you all that the SHINE program, Stimulating Hematology Investigation New Endeavors, is out there for you. We have 2 flavors, SHINE 1, which started back in fiscal year 2010, which invites topic-specific applications. The 2 that are probably most relevant to this particular group is the role of macrophages in blood cell development, and one that we call metabolic modulators of hematopoiesis. And then SHINE 2 was started back in 2013. This is a program that invites just a single aim RO1, which allows folks who have smaller scope, but still very solid preliminary data to apply for the SHINE 2. And for both of these, when you see the program announcement, or PAS, I'd like you to remember that S like a $sign. What that means is that we have set aside funds that we can use to reach beyond our published pay lines to support research that we believe is really needed for the field. So I know I've provided quite a bit of information and blown through our alphabet soup of program announcements here at the NIH, but there are actual people who would really like to talk to you about your science. Danny Gossett is our hematologist for small business, and then Shilpa Dangadi, who I mentioned at the beginning of the talk, is taking over our program on stem cell biology, bone marrow failure, and chromatin regulation. And then I'm the person to talk to for erythro and myelopoiesis, globin regulation, heme biosynthesis, and iron metabolism. So I can take maybe just 1 or 2 questions, but I know you all are really here to hear from Laurie. So I'll go ahead and stop. Thanks.
Thank you so much, Cindy. Are there any questions that anybody wants to ask? I don't see anything populating, and so we'll move on. Today, our speaker is Laurie Jackson, who's the director of the Iron and Heme Corps. Laurie got her Ph.D. at UT Southwestern, where she studied in the laboratory of Margaret Phillips, and she studied ornithine decarboxylase in very enzymatic detail. So she's a very trained biochemist. Her degree was in molecular biophysics.
Talk is now beginning.
Okay, I will be there. Oops. Let me just ask and remind everybody to please mute themselves and also turn off their videos during the presentation. If you have questions, please populate them into the chat and I will moderate that, ask questions. And then also at the end, we'll have an opportunity to ask questions of Laurie. So with that, Laurie, it's all yours. Thanks so much for presenting today.
Okay. Thank you, Diane. So whether you're watching live with us right now or whether you're watching this as a recording, the purpose of this talk today is just to help you understand how the iron and heme core can help you with your research. So the Iron and Heme Core was set up specifically for the Center of Iron and Heme Disorders in late 2016. And since that time, we've served 34 different laboratories at 20 different research institutions. And I just want to thank all those of you who have remembered to acknowledge our work in your scientific publications. We really appreciate it. So there are two of us in the Iron and Heme Core team. This here is Hector Bergonia, and he's in charge of all things heme. And I'm in charge of metal analysis. So today I'm going to talk to you about what we do, how we can help you address your research questions, and then I'm going to give you an example of some work done by Hector for a client study. And then finally, we'll talk to you a little bit about what you need to know in order to submit samples for analysis. So anybody watching this, I don't have to tell you how important iron and heme are to life. This is the figure shows the canonical heme biosynthesis pathway and catalyzed in 8 different steps. The first committed and rate limiting step is the production of ALA. 2 ALA are condensed to form the pyrrole porphobilinogen. 4 of these molecules are oligomerized, and then this is followed by ring closure by modification of some of the side chains by decarboxylation. And then finally, protoporphyrinogen 9 is oxidized to form protoporphyrin 9, and then iron is inserted into the macrocycle to form heme. And I'm going to be using this figure here just to show you what some of the services that we provide in our core. So one of the things that we do in our core is we help you analyze the activity of all 8 of these enzymes. So all the way from the first rate limiting step, catalyzed by ALAS, to the final step, catalyzed by ferrochelatase to insert iron in to form heme. And most people are actually requesting ALAS and ferrochelatase. Those are the most popular, but we can help you with any of them. This past year, we've helped people with uroporphyrinogen synthase and protoporphyrinogen oxidase. So in addition to helping you with enzyme assays, we also do metal analysis. Since we're under the umbrella of the Center for Iron and Heme Disorders, most of our users that know about our core are interested in iron. And we primarily do iron, copper, zinc, manganese, but we've helped people with many other elements. And at the end of this talk, I'm going to talk to you a little bit about what the capabilities of this instrument is and what we can help you with. So in addition to enzyme assays and metal analysis, we also help you analyze and quantify levels of heme and heme biosynthesis pathway intermediates. And in the pathway, I've circled some of the most frequently requested services. People request that we quantify this intermediate aminolevulinic acid, or ALA. We also frequently have the request to quantify porphyrin intermediates. And what these are is anything from uroporphyrinogen to copper porphyrinogen and all the different decarboxylated intermediates in between. And for those of you who use our services now, we used to have a service called porphyrin analysis, and this was a combination of porphyrin intermediates and heme and protoporphyrin 9, but we've now separated that into 2 services because the extractions are different, the separation protocols are a little bit different, and just to simplify things, we do that. So just know that you need to order those separately. And also, if you're interested in the 1 and 3 isomers for the intermediate porphyrins, which are controlled by this enzyme, U3S, we've now made it possible to directly select those. But whenever you select that, please let us know because it's actually a different protocol to separate those isomers. And then finally, one of the services that people ask for is heme and protoporphyrin 9. And this is by UPLC analysis, as were these. And you should know, if you're asking for heme and protoporphyrin 9, that it's primarily porphyrin or heme B that you're looking at. If you're interested in quantifying hemes A, B, and C, we also have a spectral analysis that we can do for you. And if you have any questions, please contact us. We're very happy to talk to you about how we can help you. So some people ask us, you know, they'll say, we believe that heme biosynthesis is affected in our system, but we don't know what step. What can we do to figure out the step? So the first thing we recommend is that you analyze heme and the various intermediates, and then look for increases or decreases of these in your perturbed versus your wild type system. And then once you understand what may be perturbed, then you can hone in, and we can help you with measuring specific enzyme activities. And then finally, if you believe that you have defect in iron levels, we can also help you with that. So next, I'm just gonna give you an example study to just kind of show you what the behind the scenes work is for a couple of these different assays, and show you how this resource has been applied to enhance iron and heme center research. And I'm going to be presenting in a very simplified way some work done early on in our core facility. And this work was done by the late Barry Pa and Yvette Yin. And I believe when she did this work, I believe she was a postdoc. For the last few years, she's been an assistant professor at the University of Delaware. And it's Hector Begonia from our core who helped them with their analysis. And basically their paper describes a study where they're understanding the basis for a disorder caused by a defect in heme synthesis, and that's EPP. So EPP is erythropoietic protoporphyria. And porphyrias, I didn't realize I hope you can see it. Porphyrias are basically caused by defects in heme biosynthesis. So the last 7 steps in heme biosynthesis, if any of these are impaired, you're going to get buildup of intermediates, or if you have an upregulation of ALAS, you'll get a buildup of toxic heme biosynthesis intermediates. So in this particular disorder, EPP, it's characterized by an accumulation of protoporphyrin 9, and this leads to an acute painful skin photosensitivity. And 90% of the people with EPP have a defect in ferrochelatase, and that leads to the buildup of protoporphyrin 9. But a certain number of them have a gain of function mutation in ALS, and since it's the rate limiting enzyme, it's going to really drive the heme biosynthesis and overwhelm the capacity of the cell to deliver iron or overwhelm ferrochelatase activity, and it'll lead to the accumulation of protoporphyrin 9. But about 1 to 5% of people with EPP have no ferrochelatase or ALAS mutations. And this leads people to search for other genes that may lead to this disorder. And in the paper that I'm going to discuss today, they describe a mutation in the CLIP-X that leads to EPP. So they identified a case of EPP of unknown etiology, and it's early onset, and it led to acute painful phototoxicity as well as the expected elevated protoporphyrin 9. And the father and the uncle also had elevated protoporphyrin 9. But no mutation was identified in ALAS or ferrochelatase, and so they did a linkage analysis in whole exome sequencing. And they identified a single nucleotide substitution that led to a glycine to aspartic acid mutation in the affected people. And this was inherited with Mendelian dominance. And they proposed that this CLIP-X mutation increases protoporphyrin 9 in these individuals. So briefly, just a little brief background. CLIP-X is a mitochondrial unfoldase, and it's ATP dependent, its activity, it's hexameric. And CLIP-X interacts with CLIP-P. So ClipX functions as a hexamer, and it interacts with ClipP, and it forms a protease complex where ClipX will bind and unfold proteins in an ATP-dependent manner, transfer it through the proteolytic chamber, and those proteins will be degraded. ClipX has also been reported to bind and activate ALAS through its unfoldase activity. And it does this by assisting it binding to its cofactor, PLP. And this mutation that they identified is located in the highly conserved domain that's important for ATP binding and hydrolysis. So just very briefly, the investigators first verified that this mutation does impair ClipX ATPase activity. And then they created a series of experimental models. So either, looking at housekeeping, heme biosynthesis, and HEK293 T cells, or erythroid in male cells. And each of these cells are overexpressing flag tagged either wild type ClipX or mutant ClipX. And they also used an animal model of cRNA injected zebrafish embryos, again, with ClipX, wild type, or mutant. And the iron and heme core assisted them by addressing the following questions in these experimental models. They used experimental models to validate whether ClipX was involved in EPP. So does this mutation increase protoporphyrin IX production? And does this mutation alter heme biosynthesis? So first, Hector helped them measure protoporphyrin IX levels in their samples. So what happens behind the scenes when you provide us with samples? So this is Hector working away using his HPLC. What happens is we typically, and actually for all cell pellets, for all of Hector's assays, he requires at least 100 microliters of packed cell pellet, irregardless of the type of cell you're sending him. That's what he requires. And I'll give you a little bit more information later about different samples that he'll ask for. So basically what's going to happen is he's going to lyse your cell pellet, and then he's going to quantify the protein concentration in those lysates or homogenates using a protein assay. And then he's going to adjust all the samples to a set concentration, and then he's going to extract, in this case, heme and protoporphyrin IX. And in this case, it was using ethyl acetate and acetic acid. And then he's going to inject those extractions over his ultra-performance liquid chromatography system. So this is an example of a chromatogram from his UPLC analysis where it's run over reverse phase. And protoporphyrin IX is fluorescent. And this is the excitation and emission wavelengths here. This is fluorescence, and this is elution time. And this is the protoporphyrin IX peak. We took just a couple of example peaks from the Jens study. Hector still has them. And basically over here is just a close-up, and these are just overlaid spectra. This is the background. This is pretty nice background. And then these are just 3 different samples that he selected that are 2-fold or 3-fold different from each other in concentration. And one of the things that we want to bring up for those of you who use our core is that many of the intermediates are near the limit of detection under normal conditions. These are not intermediates the cell wants to build up. And so it's really important that you send us the amount of material that we request. This group is really good about giving us sufficient material. And we just want you to know that the sample amount that you provide is going to impact signal to noise and data quality. And so please send us sufficient material and replicates. So basically from analysis like this, Hector is going to return picomoles of protoporphyrin IX per milligram of protein. So everything's normalized to the protein. So once he returns this, the investigators have done multiple experiments. They process the data, they plot it. And this is actually taken from their paper. This is HEK293 T cells. These are the male cells. This is amount of protoporphyrin IX, picomole per milligram protein. These are control cells. These are cells overexpressing ClipX or overexpressing the ClipX mutant. And what they see is they're seeing an increase in protoporphyrin IX when you overexpress the mutant in these cells. And increased protoporphyrin IX is consistent with the EPP phenotype. So in EPP, like I said, it's characterized by the buildup of protoporphyrin IX. And that can be due to either decreased ferrochelatase activity or increase in ALAS activity. And as I mentioned earlier, ClipX is known to bind and interact with ALAS. So they next asked the question, does this mutation lead to altered ALAS activity? And once again, Hector was able to help them with their samples. So just in a nutshell, what's going to happen here is Hector is going to lyse the cell pellets and once again quantify the protein and adjust the sample concentrations. And then he's going to mix these lysates, whole lysates, with the different substrates. And he actually synthesizes succinyl-CoA in laboratory, as well as the cofactor pyridoxal phosphate. And then this is going to produce ALA, and he's going to stop the reaction, and he derivatizes ALA to form a fluorescent product, which he can monitor by UPLC analysis. And this just shows one example chromatogram with a fluorescent ALA derivative. And then he is going to return your results in nanomoles of ALA per milligram protein per hour. And I said this really quickly, but this is a lot of work for him. So these are just two example traces similar to the last one, showing, you know, here's the peak for ALA, and then here's a zoom in. And he, once again, just pulled a couple of traces from this analysis. And the ones he happened to pull, this is background, it's nice background. And this particular sample is 1.3-fold higher than this one. And like I said, he returns the data in nanomolar ALA per milligram protein per hour. And once investigators get this data, so here they plot it, like I said. So here's HAK293 T cells, male cells, and zebrafish embryos with either wild type or mutant overexpressed. And he's able to show, or they're able to show, increases in ALAS activity when you overexpress the mutant ClipX. So remember, increased ALAS activity would drive heme biosynthesis and could lead to protoporphyrin IX accumulation. And this is consistent with the EPP phenotype. And the investigators went on to show that the activity correlated with increases or decreases in protein level. So this again, just repeating what I said, they see the increased protoporphyrin IX, which leads to skin photosensitivity in their model systems. They show increased ALAS activity. And because at the time it looked like ALAS is affected but not ferrochelatase, it suggests the possibility that it could be either limiting in ferrochelatase or iron. And they suggested that iron treatment could help patients with this form of EPP. And it's nice to see someone be able to apply it or find ways to suggest ways to use this information. So that was the end of the Iron and Heme Core work for their study. But they went on, I'm just going to say really briefly, it's a nice study. They went on to verify this phenotype in cells that were derived from the patients. And then they went on to investigate a little bit more, how does this happen? And they demonstrated that ALAS2 degradation is impaired in these cells, and that's ClipX dependent. And they're able to show that the mutant ClipX, shown in here in red, does co-assemble with the wild type ClipX, but this mixed, what is it, hexamer? Oh no, interacts more weakly with the ClipP, and so you get less degradation. And because you're getting less degradation of this protein, this is ramping up ALAS and hence heme biosynthesis, and you're getting a buildup of the intermediates. So in summary, they described a new molecular cause for EPP. And the Iron and Heme Core was able to provide assistance to this group by measuring protoporphyrin IX and ALAS activity. And Yvette Yin is now an assistant professor, and she's a CIHD member, and she continues to use our core. And she's told me that she's submitting a paper, getting ready to submit a paper to look, it's a study of more in depth to how ClipX is regulating erythroid heme synthesis and controlling mitochondrial heme synthesis enzymes. And I look forward to that paper coming out. And she uses our core even more extensively than she did in the past. And she's requested multiple different assays, enzyme assays, intermediate assays, as well as metal analysis. And a lot of her work for her new publication has been supported by CIHD pilot funding. So it's kind of really nice to see everything come together. So what do you need to know for sample submission? So in addition to these set assays that we've told you about, we will work with you to develop an assay to fit your research needs. So for example, Hector has about 25 years of experience with these enzyme assays. And so he's used this to help various investigators. So for example, until very recently, we had about 7 of the 8 that were really ready to go in our laboratory. Protoporphyrin protoporphyrinogen oxidase, it's much more challenging, and so we needed to get it, just tweak a few more things to get it set up in our laboratory. But as soon as Dr. Yan told us she was interested in it, Hector got right on it and got it ready to go for her laboratory. We've also recently, as another example, helped someone. So normally we would measure ferrochelatase activity, where iron is inserted into the macrocycle to form heme. But this group is a microbiology group, and they were interested in an activity where the iron is removed from heme to form protoporphyrin IX. And so Hector was able to develop this assay, and it's led to a couple different publications, and we're pretty proud of that. Also, just in general, he has so much experience. If people are interested in porphyrins and flavins, for example, he'll do this for them. He's recently working with another group. He's going to add microbilin analysis onto porphyrin analysis. But if you're interested in something, please call us. That's one nice advantage of being a small core. We can work with you. Our most important recommendation is please send sufficient sample. So, gosh, I'm talking kind of fast. Sorry about that. But this talk's probably got about 15 minutes left. So please send sufficient samples. So I've already shown you examples, but Hector gave me some sample traces. These are UPLC traces for intermediate porphyrins. This is uroporphyrin, and this would be copper porphyrin, and these are the various intermediates here. 7, 6, and 5 carboxyporphyrin. And you can see these intermediates aren't meant to build up real high in cells. And so in an unperturbed sample, you may see really low levels. And these are just more traces of actually perturbed samples. Just so you understand, a lot of people, we ask for 100 microliters packed cell pellet, and each time they kind of send us less and less and less. And the signal, it's important to send us enough material. We're also able to, you know, if we have really large signal, we're also able to separate it very nicely. So I keep showing you these small traces. But so please contact us if you have any question about material required. I hope you can see this on your screen. We have our web page is right here. We provide information about how to prepare your samples and how much material. But really, right here, for all of Hector's assays, so the enzyme assays, the heme assays, the intermediate assays, he requires about the same amount of material. So if you're sending him a cell pellet, he requires about 100 microliters of packed cell pellet. If you're sending him tissue, he requires 100 milligrams of tissue. Or if you're sending a lysate, he requires about 1 milligram of protein worth of lysate. So 100 microliter of 10 mg per mil protein homogenate or lysate. So please send sufficient sample. So as I mentioned, I help with metal analysis. Let me see what time it is. And I do this using an Agilent 7900 ICP-MS. And the most important recommendation I'm going to be giving you here also is please send sufficient sample. So ICP-MS is inductively coupled mass spectrometry. And basically, an argon plasma decomposes and atomizes and ionizes your sample. And then the elements are identified based on mass charge. And this is useful for the majority of metals and nonmetals. And I like to use this figure by Perkin-Elmer of the periodic table because it just visually is a good illustration of what elements ICP-MS is good for. It's not good for the white, but it's very good for the blue elements and the yellow elements. And it can also help you with these other elements here. They're just slightly less sensitive. So most people in our core, as I mentioned, request iron, manganese, copper, and zinc. But we've helped people with all sorts of elements, and we're happy to do that. And we have a different series, several different stock solutions, calibration solutions, and you just ask us, tell us what we want. We'll tell you what we have available, and if we don't have it available, we'll order it in. But yeah, feel free, just contact us. We can talk to you about what we can do for you. So the metal analysis services, we can take it all the way from just giving us a sample to the analysis. So if you give me a cell pellet or tissue, most of our investigators, there's different ways to normalize this data. Most of our users normalize to protein concentration, some normalize to cell number. If you're interested in normalizing to protein concentration, I will either homogenize the tissue or the lysis cells. And then, like Hector, I'm going to use the protein assays for normalization purposes. Then I'm going to digest your sample, and I use hot block digestion, where we digest with nitric acid and hydrogen peroxide. And then we dry down your samples, resuspend them, and then I do metal analysis by ICP-MS. So for those of you, for everybody in the CIHD core, you don't pay tech time. That's one of the advantages. So it's much cheaper for you. But if you're not a member of the CIHD, I want you to know that it's a lot less expensive if you, it's not going to affect CIHD members much, but if you're not, it's less expensive if you homogenize and you run your protein assay prior to sending your sample to us. But if you do this, please make sure that you use pure water or PBS. And when you do this, please send a sample of your water or your PBS. At least a ml, more is better for a blank, so that I can determine what your background's like. And also, if you send me a cell pellet, if you've washed it with PBS, which you need to do, please send me a sample of your PBS so that I can measure background. So determining the amount of sample to send for metal analysis is a little bit more complicated than Hector's, because it differs quite a bit for different cell types. If you're sending me and different tissue types. But basically, if you send me at least 100 milligrams of tissue, that's great. I wouldn't go much lower than 25 milligrams of tissue, although some people have sent me less and it's worked, but just communicate with me to make sure that's the sample that I've worked with. And if not, I can help you with the pilot study that I'll describe in a moment. But please feel free to contact us with cell line specific information. This table here is just an example of different cell lines that we've tested. And we're looking for about 20 parts per billion iron on the instrument. Our method detection limit is 1.3 parts per billion. So we would like people to be at least 20 parts per billion on the instrument in order to have good data, and that's minimum. We would like you to send more if you can. So basically, like I said, it's going to vary widely, the amount of iron that you expect to be in the cell lines based on what the cell does, how much iron they typically have in them, what the cell size is, and that type of thing. But if we don't have information about your cell type, we will run a pilot study for you. And I do this for people all the time. Don't hesitate to ask. You'd basically send me a couple different concentrations, and I'd start with maybe 10 million cells, 2 million cells, 5 million cells for your wild type and your perturbed system, so that we can get an idea of what the signal is. So we can, before you set up an expensive study, just make sure that we're going to be able to get you the type of signal that you need. And like I said, the amount that I'm recommending here will be a minimum amount. Please send more material if you can. And this is an example of why I say send more material. And it's just helping you understand one of the challenges we see with ICP-MS data. This shows 13 different blanks that I ran for one run and a couple different data sets. And each of these data sets, the on-instrument iron concentration was 17 parts per billion for both of them. But what you can see here is I typically, for my blank, I see less than 1 parts per billion iron. That's pretty normal. But occasionally I see these outliers. And maybe a piece of iron was on a tip, in a tube, in the water, in the air. But we can see this kind of spike when we're close to background. And you can see with this data here, as you get down lower, you approach this noise that we see in the background. So if you can send us more material and we can get the numbers higher, you're going to be less, your data is just going to be more solid. And so please send more material if you can. Replicate experiments are recommended. So users can also create their own homogenate, and I'm not going to go through the amounts here, but we know from experience what kind of readings people have been getting with what type of sample, warm homogenate, mitochondrial isolated from different culture cells versus tissue, and just kind of what to expect. And like I said, we are very happy to run a small pilot study. We do it for free on the back of somebody else's experiment. We just put it at the end on when we do an ICP-MS run, and we're happy to do that for you. But please, when you give us cell pellets, we don't want to measure the iron in the media. We want to measure the iron in the cells. So please wash the cell pellets, remove all the PBS, that's important, and then send us a blank, and make sure you send us plenty, at least a mil, more is better, and then ship the samples to us frozen, or deliver it to us frozen. So for these are some of the different elements that we've measured. Like I said, background for iron is 1.3 parts per billion. Sorry, method detection limit. But manganese is much lower. It just depends. Iron is everywhere. It's going to be in the plasticware. It's all over. And so the detection limits are pretty good, but still higher. So each time you submit a sample to us, particularly for metal analysis, please send a list of sample names and what elements do you want measured. So and make sure you tell me each time how you're going to normalize your samples to cell number, protein concentration, weight, and what's going to be done by the core and what's going to be done in the lab. Have you already done the protein assay? Do you want me to do the assay? And if you don't need a protein assay, I'm going to directly digest the material. So thank you so much for your time. Apologize for talking so fast. And thank you to all the people who cite our core. And on our website right here, CIHD course, Utah Edu/iron and heme. There's all the information I described and more. It's also our contact information, as well as how to cite the core. So thank you so much for your time. I appreciate it.
Yeah, thank you, Laurie, very much. We don't have any questions populated in the chat. You were either very clear or we have no idea.
Okay.
But it seemed clear to me. So does anybody have any questions? If you do, you're welcome to unmute yourself and ask.
I have a question. You asked about Jake.
Go for it.
Thank you very much for this presentation. And I have just one question. I think one of the major problems we have to address with iron is absorption to cells. And so I think that many times it's highly recommended before one does a lysis or homogenate is to remove any extracellular, not extracellular, but material which is absorbed on the surface, because in some of the cells, this contamination could be as high as 60%.
Wow, okay.
And this is something that I have experienced myself quite a lot. So I think this is a caution that I want to say to anyone who is measuring or thinking that it's measuring it's cell associated, but not necessarily internal iron.
So what would you recommend in addition to PBS washes?
I think that one wash with some sort of a chelator, which is non-penetrating, like DTPA or something of that sort.
I will test that. Thank you.
Tom Gantz has a question. Tom, do you want to unmute yourself and ask?
Yes. Do you do isotopic analysis of metals?
Well, so when you say isotopic analysis, what do you mean?
So if we have cold isotopes and we label our cell cultures, does your facility distinguish between the isotopes? Can you use a mass spec to distinguish between the isotopes?
So I don't think that we look at single isotopes. That's what we do. But so there are certain instruments that are set up specifically to do that. I would have to check. It would depend on the isotopes, because part of the problem when you have something that has all the metals in them is there are multiple isotopes for each element. And so you have to work at isotopes where the different elements aren't going to overlap. And when you run the analysis, and so I'd have to be able to check your particular isotopes that you're looking at to see how well that would work on an instrument. I do know that, yeah, I would have to, there's another group I know that does that, and I would have to talk to them. I haven't done that myself. Okay. But it would depend on the isotopes you're interested in. So.
Thank you.
I have a question.
Go for it, Paul.
So is there any chance that Utah, or maybe with the help of Cindy, get a laser ablation ICP-MS?
I would love
that we can actually have a good analysis on a solid sample instead of homogenizing, because homogenization does not help with any kind of subcellular compartmentalization of metals, right?
Right, right. It would be nice. It would be wonderful, but there would have to be funds for it. We'd have to be able to show that a lot of people were interested in it, I imagine.
Because the one on our campus is run by geologists, and they don't want any biological samples coming in, right?
So I'm going to chime in there and just say, if there are people on this Zoom who are interested in that type of thing, please let Laurie know so that we can kind of get an estimate if that's something that we maybe need to look at trying for new instrumentation.
And maybe Laurie, you can describe what LA ICP-MS does, because it's really powerful. It gives you a spatial perspective of where the metals are.
I love that.
Yeah.
Anyone else have any questions? You're welcome to all unmute yourselves and put your videos on to be able to say hi to all our good friends here who are interested in iron and heme. And Laurie, thank you again for your presentation. I'm very curious. I wish Yvette was on. She's not on. I'm very curious about why you accumulate ALA and just don't pump it through if your ferrochelatase is working fine. Are you iron limited? Are you ferrochelatase limited in her ClipX mutant?
So she's her next paper is going to talk about that.
Good.
Great. Yes. So it does. I don't want to tell you what's in the paper. I've read it. I think she should do that. So yeah.
I agree. I wish.
Okay. Okay. Okay.
I'll ask her next time.
Okay.
Anyone else? Okay. Thank you all so much, and we want to thank Cindy again for talking about things that are available at NIDDK and the hematology program. And wish you all well, and we'll see you in another at another session in December, where I think it's Iqbal Hamza who's going to talk to us on the 8th of December, if I've got the date right. Check your schedules. I don't have it in front of me, but thank you very much. And if you have suggestions for the core, reach out to Laurie, and they're great. I hear them talking all the time to people who are running samples through there. So thanks again, everybody, and nice to see everyone again. Hi, Jan. Hi, Stefano. Hi, Victoria. Hi, everyone. Very nice to see you all. Everybody stay safe. And if you have further questions at all about the CIHD here at Utah or the U54 mechanisms, please reach out to me. I'm happy to help you. Bye, Tom. See you.
Bye-bye.
Okay.
Hi, guys.
I keep looking at everybody's names, wanting to wave at everyone. So nice to see you all. Okay. All right. Thanks again, Laurie. And thanks again, Cindy. I'll end the meeting now unless anybody else has other comments. Great. Bye-bye.
Bye.